Prophase 1 starts with a diploid cell. Its chromatin contains two uncoiled, spread-out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of four sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform.